Hey team, this is going to be the last video before the first test. So this is going to be a short video. All we're going to be covering is this idea of how water moves in the phloem and how how water moves in the xylem and how sugar moves in the phloem. I actually did this in the last video. I had it all done, except like a dummy, I forgot to share my screen with you. So it was just me talking the whole time and you couldn't see anything I was drawing. So I'm gonna have to redo it, but that's okay. I'll get the video done, and then we can even take a little bit of time to talk about the test and kind of what you can expect on the test. And so let's finish up. Let's think about for a second how stuff moves in general. There are some certain laws about how things move, like the law of diffusion, which says basically that stuff moves, and by stuff we mean molecules in any state, from higher to lower concentration. And, you know, in science, if something is a law, that means it always happens, right? There's no exception to the rule, okay? There's also the law of osmosis, and osmosis specifies what's moving. It says that water moves by diffusion through a semi-permeable membrane. You probably remember these definitions from your general biology. So, you know, what does this mean? Well, what's the semi-permeable membrane? It's the cell membrane, right? That's what we're getting at here. And it turns out that if you have a concentration gradient for water, water will pass readily through this cell membrane. The membrane isn't really a barrier to water. So let's say we had a cell that was 5% salt, and we'll just do this as an example. And we bathe that cell in a solution that's 10% salt. And then I might want to know, which way is the water going to go? Is it going to go into the cell or out of the cell? Well, the first thing you have to realize is that the water doesn't move. The salt doesn't move. The salt is stuck wherever it is. So the salt inside can't get out and the salt outside can't get in. What moves is the water, and it moves by diffusion. So it's going to move from a higher to a lower concentration. Well, if there's 5% salt, in here, there's 95% water. Out here, there's 10% salt. So there's 90% water. And now you can see, okay, the concentration gradient says water is going to move from higher to lower through the membrane. So water is going to actually leave the cell. Okay. You know, we could show one at equilibrium, you know, 10% in and 10% out. And then, you know, the water's not going to move. It's at equilibrium. It's not going in or out. We could do the opposite scenario with a cell. We could say, okay, we got 10% inside and 5% outside. So that's 95% water on the outside and 90% on the inside. So the concentration gradient is going this way. So osmosis is a law, and it's actually how a lot of stuff can get in and out of the cell. It's just the reality of it. Now, if you want to force something into a cell against a concentration gradient, you have to use energy, right? And that's called active transport. But just in general, stuff will move in and out of cells based upon diffusion or osmosis. Okay, so those are just more general ways that stuff moves. What we want to understand now is, how does water move up the xylem. Remember, xylem is a one-way street. Now, if you think about the tallest tree you can think of, around here, that's going to be probably a tulip poplar or a white pine. And some of the tulip poplars out at Joyce Kilmer are 150 feet tall. Okay. So what we need to be able to explain is how to move water up, up to 150 feet around here. The problem is that the xylem is not living. These cells aren't living. And so it's a little bit of a challenge to explain how you can move something without energy, right? Because there's no chemical energy to move the water because the xylem isn't alive. So the theory is called the cohesion tension theory. It's a difficult thing to prove, right? But the cohesion tension theory tries to explain how water moves up the xylem. And the best way to really understand it is if we draw a tree, this, and here's a leaf. Now I'll draw some xylem vessels. So hard to draw on this thing. It's hard to draw a straight line, but I'm going to do my best. Well, that's pretty good, huh? Look at that. So what this represents, not to scale, is the xylem, right? All these xylem cells stuck together like soda straws from the roots all the way up the stem and to the leaf. So here's what was previously thought. One of the things they examined was what's called osmotic pressure as a possible way that the water moved. They said, yeah, there's all this water in the ground. You have a lot of water, and that creates pressure for the water to move in. And so the water wants to move then, but from this higher concentration to this lower concentration based upon osmosis. And so they did some calculations, and they determined that osmotic pressure would in fact move the water about 3 feet up to the xylem. And that's not quite enough. We need to explain further movement than 3 feet. And so then they said, well, okay, that's part of it. The other thing is capillary action. We know that these are very narrow diameter tubes, essentially. And we know that water is polar and it sticks to itself, and it also will stick to the inside of the xylem cells, the cell walls. And so they did some calculations. They measured the diameter of the xylem. It's a very narrow tube. And they said, yeah, it will in fact move the water. Capillary action will move the water about 3 feet. So far we've moved the water 6 feet, right? So then they came up with this theory, and it's called the cohesion tension theory, that the basis of the theory has to do with the fact that water is polar, right? Water sticks to itself. It has cohesive properties. Cohesion means it sticks to itself like this. Everywhere you have a hydrogen of one water molecule, it's attracted, this is a slightly positive charge, it's attracted to the slightly negative charge of the oxygen, et cetera, et cetera. So the theory goes like this: when the plant is first born and it's very small and short, a water column forms. And so we have all these water molecules in a water column moving up the xylem. And what the theory says is what's holding these water molecules together are hydrogen bonds, right? Hydrogen bonds, it's like they're all holding hands in a sense. And so here's what the theory says. As this water column forms, as one water molecule evaporates from the surface of the leaf at the top of the plant, another water molecule is pulled in to the xylem at the bottom. And so it's a one-to-one exchange. For every water molecule you lose at the top, you get another one at the bottom. And as long as they keep holding hands, then this water column can grow with the plant, essentially. Okay. There's a word for the water evaporating from the surface of a leaf, and that word is called transpiration, and I want you to know that word. What is transpiration? It's evaporation in plants. It's the evaporation of water from the surface of the leaf. Remember we talked about the stoma, those little pores, usually on the underside of the leaf, and that's how the water transpires. That's how it leaves. So plants are constantly losing water through transpiration, assuming that their stoma are open. Okay. Now, one thing you may think about is a plant will die if it runs out of water, right? And we can explain that. There's this thing called the permanent wilting point. And you may have experienced this, I hope not, but you may have experienced a permanent wilting point in houseplants where maybe you went out of town, you forgot to get someone to water your plants, and you come back and they're all just wilted. And, you know, sometimes it's like a miracle. You water them and they just, within an hour or two, they perk back up. But there's this point at which they will not ever come back. And we can explain why that is. What can happen is as the water starts to run out down here, it's still evaporating from the surface of the leaf. It creates a tension. That's the tension part in the cohesion tension theory. Creates a tension in this water column. There's always a certain amount of tension in it. But if there becomes too much tension, the water column somewhere in the middle will break. And when that water column breaks, these water molecules just kind of dribble back down to the bottom. These evaporate off to the top, and now we have a hollow tube. And there's no way to ever reestablish that water column in the plant, and the plant dies. So you want to be sure not to let your plants reach the permanent wilting point. And, you know, it's a trade-off for plants with moisture and growth because they need water, right? Okay, so that's the cohesion tension theory. And again, this tries to explain how the water moves up the xylem. Okay, what about phloem? How does sugar move up or down? Because xylem is a two-way street. Phloem is a two-way street. And it's important to understand that the sugar is dissolved. I'm going to put dissolved just to remind us that the sugar is not moving as a solid sugar molecule. It's dissolved in water. It goes into solution in the water. So let's draw our tree again. It was such a pretty tree the last time. Let me see if I can do as well this time. This might even be better tree, y'all. Look at that. Look how pretty that tree is. Okay, I'm getting better at drawing this thing. And then I have to do now I got to do the same thing with the phloem. I got to draw sieve cells. And remember, they're all stuck together kind of like soda straws as well. And we'll show it actually passing into the leaf because it does. Okay, that's pretty good. All the way down to the roots. Okay. How does the sugar water move? The theory is called the pressure flow hypothesis. And here's what it says. Now, remember, phloem is alive. We can use energy now because phloem is alive. Here's what it says. The pressure flow hypothesis says that the sugar moves from source to sink along a concentration gradient. Let's think about what that means. The source is where the sugar is, and the sink is where it needs to be, okay? You can't always assume that the source is the leaf and that the sink is the roots. It really depends upon the time of the year. For example, in the late winter, where's all the sugar in the tree? It's in the roots. It's being stored for the winter. There's this thing called the sap flow, where the sugar starts to rise up from its source, the roots, up toward the developing buds of the tree, right? So in the wintertime, really the source ends up being the roots and the sink ends up being the buds that are developing to become leaves. However, during the growing season, spring and summer, the source of the sugar is in the leaf because that's where it's being made. And we're going to let all these little triangles represent glucose molecules. Okay, that's glucose. And here's the deal. We have a concentration of sugar here. And what we're going to do at the source is what's called active loading. Active loading at the source. What that means is we're going to actively move the sugar from the leaf into the phloem at the source. Okay. And you could imagine that this isn't free, right? We're moving these sugar molecules through membranes and it's going to take energy to do so. Glucose is a large molecule. And if you remember what the energy source is for these types of reactions, it's ATP, right? It takes one ATP molecule. So I'm going to show ATP being converted to ADP. That's the energy source that allows the active loading of the sugar molecules at the source where they are, okay? Now think about what's gonna happen when we do this. We're creating a concentration gradient in this tube, okay? Is the glucose, each molecule just gonna sit there? No, the law of diffusion says this stuff is now going to flow, it's going to diffuse. And so it's going to go from a higher concentration to a lower concentration. So here it comes down the tube, right? And eventually the sugar, some of the sugar arrives down here at the roots, which is the sink. And guess what we're going to do here? We're going to actively unload the sugar. So we're going to say active unloading at the sink. Think about what that does. As long as you actively load it at one end and actively unload it at the other, you maintain a concentration gradient, don't you? And as long as you maintain a concentration gradient, most of the work is done for free based upon the law of diffusion. So the bulk of the movement occurs just by diffusion, but you do have to put some energy in on both ends to load it and then unload it in order to keep it flowing. This is called the pressure flow hypothesis. And, we're using energy to explain how the sugar water moves. Now, if you think about the maple farmers, well, they take advantage of this, right? They tap into maple trees and they tap into the phloem and they siphon out a little bit of that water, of that sugar water rather, and then they cook it down and make maple syrup. Yum. Don't get sucked into thinking the source and the sink is the source is always the same place. It's not. It just depends upon what's going on in the plant at the particular time. And that, my friends, is the end of material for test 1. So now you are essentially ready for test 1, and I'm going to tell you a little bit about test 1. What I'm going to tell you about it is the kind of the format and what to expect. The test is timed. You have 50 minutes, and that should be ample time because it is 50 multiple choice questions, I think. There could be, there might be a discussion. I think there's actually not 50 multiple choice questions. The test is worth, I'm sorry, let's be reasonable here. You do have 50 minutes to take the test. It consists of multiple choice, matching, and discussion. That's more correct. There's a lot of multiple choice and matching, and I think there's 1 or 2 discussion questions on the test. Okay, so just be prepared for these types of this type of format. You should have plenty of time to take the test. Here's the thing, though, y'all. Once you start, like we said before, you got to finish. So don't start and then have some other obligation. So you need to start and finish in 50 minutes. Okay. You must have reliable internet. You know if your internet's reliable or not. If you're constantly getting kicked off, if you have dial-up, that is not reliable internet. So if you don't have reliable internet, don't fret. Just come on to AB Tech campus, and we will hook you up with reliable internet here on campus. You are able to come and use the computer labs, or they have laptops you can use at South Campus. If you're there, if you're close to South, I would recommend just coming to Victoria Road. I know we have computer labs set up. Check with student services about the locations of those. Okay, and at this point, you know, once you've seen this video, you have gotten all the information that you need to study and do well on the first test. So I'll be opening that test up. That test probably will be open sometime after you watch this video. And I will send you a course announcement letting you know when it does in fact open. Oh, the other thing to tell you, typically it's open for 1 week. And that just means you can take it any time during that week that it's open. But guess what? It never reopens. Think about what that means. It never reopens, y'all. Once it's closed forever. You can never go back and take it. So don't miss the deadline. Put it in your phone. Make sure you don't miss the deadline. Okay. I think that's all I have for you. I really appreciate your helping us get through this difficult time, right? And I will be in touch. I'll let you know when the test opens. I hope you have a good night. Foot hurts. I broke my foot. I told y'all that. I broke my foot. It's awful. 6 weeks I have to wear this boot. All right. Have a good night. Bye-bye.